Welcome everyone. So today our speaker is Dr. Martina Preiner. So Martina is a chemist at the Heinrich Heine University of Düsseldorf in Germany, and also a freelance journalist. So after working for radio, television, and print media for 7 years, Martina returned to research. And so Dr. Preiner work focuses on the chemical origin of life, and today she will present her work on the serpentinizing systems and hydrogen activation in early metabolism. So just so you know, the talk is recorded. So if you have any question and that you wish that your question is not recorded, please email your question to me, and then I will forward it to Martina. Okay, Martina, yeah, the floor is yours.
Thank you very much, Thomas, for that introduction. Also very well researched, because that usually is nothing that comes to light that I used to be a journalist. So I will try to share my slides, hopefully without any problems. Yes, so it should look okay, right? If not, just let me know. So thank you very much for this kind invitation, and my best greetings from Düsseldorf to the East Coast. Hello, good morning, good evening, whatever you prefer. And I will talk a little bit, as Thomas already said, about serpentinizing systems and hydrogen activation within them and their connection to early metabolism. So the beginnings of life can be approached from many different scientific disciplines. And in order to get or to start a little bit more from my biological perspective, I usually choose to present the work of these two gentlemen, Charles Darwin and Ernst Haeckel, and especially those family trees they were drawing. So basically what they were doing is comparing existing life forms, morphologies of the existing life forms, and trying to find something out about descendants of those, and also trying to figure out where we all come from, what are our common ancestors. So when we go down from all the branches of life down to the stem, that is where we have our common universal ancestor. And when it comes to origin of life, we usually have to go below that. So below the stem, which are the roots of life. So the connection between geology and biology. What is at that stem is a theoretical construct so far still, the last universal common ancestor, or for short, LUCA. So that's something very important, so I will just repeat it again. It's really just a theoretical construct of this first half-life cell from presumably bacteria and archaea originated. And we picture it as kind of something that was still lodged into a geochemical system and very dependent of the geochemical system that it lived in. And how do you get to reconstruct this theoretical construct? It is a very typical top-down research approach, which is not my work. It was done by colleagues of mine already, oh God, 5 years ago. I just realized, over 5 years ago, where they were basically comparing the genetic setup of diverse bacteria and archaea, and trying to reconstruct the physiology of LUCA by investigating these phylogenetic trees, not unlike what Haeckel and Darwin were doing, and trying to figure out what is the most conserved part of this physiology, and then assuming that this physiology must have been or could have been the one of LUCA. And what they reconstructed was something like this. So we picture the physiology of LUCA as an anaerobic autotroph that employed a reductive acetyl-Coa pathway. So that means it was CO2 fixing, and therefore it used the electrons of hydrogen, and it also needed a vast background of transition metals, because it was very dependent on them due to all the enzymes it had to employ to provide for these pathways it had. And from this physiology, you can then think about, okay, where did LUCA actually live? Where could it get all this CO2 and hydrogen and so on? So to sum up and be a little bit repetitive, the habitat of LUCA had to be geochemically active and rich in hydrogen, CO2, or other oxidized forms of carbon and transition metals. So what could give us that? Luckily, something very interesting was found already 20 years ago by Deborah Kelly and her colleagues. Those was the hydrothermal, the Lost City hydrothermal vent field at the Mid-Atlantic Ridge. And we call them alkaline hydrothermal vents, but usually I actually prefer to just call them serpentinizing system because that's the most striking part of them, what makes them alkaline as well. So these systems could have been an habitat of the LUCA that we reconstructed, or rather my colleagues reconstructed. So what makes alkaline hydrothermal vents or serpentinizing systems so interesting? First of all, they have a steady carbon source in the form of CO2 or also in the form of carbonates. And very importantly, they provide a steady energy and electron source in the form of hydrogen, which we need to actually reduce CO2. Inside of the vents, we are alkaline, that's the name. So we have high pHs out there in the ocean. It's presumed that it had very, very acidic pH. So we also find pH gradients in there. We have a thermal gradient. So it starts down there with very hot temperatures, can go down to very low temperatures on the top of the systems. We have small cracks and even nano-sized compartments, which are ideal for concentration of compounds if we really talk about prebiotic chemistry, for example. And also, which is probably always a little bit surprising also when you think about it, we actually can find low water activities inside the crust of these systems, inside of the crust. The crust parts to do serpentinization, which means we actually have access to systems that would not hydrolyze everything that is being produced in these systems. And last but not least, of course, the transition metals. There are a lot of minerals inside these crust systems, inside of these hydrothermal vent systems that can be employed for catalysis. So, and I don't have to tell you that, and it's also not the background I'm working on, but serpentinizing systems and icy moons, so ocean worlds, are very tightly connected. And this is just a very small spread of papers that I personally know. They're very Enceladus focused, but I know that there are a lot of other opportunities and options out there as well. So that's something that really nicely connects our type of research with what is being done from the astro and space exploration side, which is very, very interesting and nicely connected. So we have the top-down approach from a biological perspective, and in order to connect geochemistry and biochemistry in LUCA, basically, so this half-alive cell, we also need a bottom-up approach, so a geochemical or chemical approach in which we then look for parallels and possible transition points from geology to biology. But for that, to understand better what we're doing there, I have to jump back a little bit more to biology, but the upbuilding parts of biology. So the focus on the so-called reductive acetyl-Coa pathway, which is presumably the carbon and energy metabolism that was in LUCA, and today is still found in organisms that live from the reduction of CO2 with hydrogen, also called anaerobic autotrophs. So this is, of course, a lot of arrows, but in the end, the acetyl-CoA pathway is a very nice, simple pathway, the most simple pathway, CO2 fixation pathway that we know of. It's also the only really exergonic one that actually creates or produces ATP, the energy currency, and does not consume it. And it is also the only linear one that we know of. So it's basically like a one-way street into metabolism. It consists of two branches. So CO2 and hydrogen can, over the methyl branch, be reacting, or hydrogen is reducing CO2 to a methyl group. And on the other side, you have the so-called carbonyl branch via carbon monoxide. that is being tunneled to this final enzyme that then creates the acetyl group of acetyl-CoA. And this is not part of the official acetyl-CoA pathway anymore, but the next reduction step would be to pyruvate. And from pyruvate, you go to sugar synthesis and also via oxaloacetate to the citric acid cycle as well. So it's all very nicely connected to the rest of metabolism. So that's basically where carbon metabolism starts for these autotrophs. And here you can also see that the reduction of carbon is kind of the prerequisite for life. And that is what for our research it's all about, basically. So if from a very chemical point of view, you just look at CO2 and hydrogen and their reaction, it's of course the start of something, but it is unfortunately kinetically inhibited. So if you just put CO2 and hydrogen into one reactor, nothing much will happen unless you apply very high temperatures. Then you would get the so-called reverse water shift gas reaction, producing carbon monoxide and water. But if you actually have the right catalyst, you can activate the hydrogen on the surface. You can chemisorb hydrides on the surface of a catalyst, and then hydrogen can be a very potent reductant for CO2 if you actually have the right catalyst at hand, which is basically anything that has transition metals in it. I mean, not completely true, but pretty true. What you can see here is a quite dated but still nice figure I find from Gerhard Ertl, who got the Nobel Prize for figuring out how the Haber-Bosch process works on metal surfaces. So the reduction of nitrogen with the help of hydrogen. And here you can see iron atoms nicely ordered. And on top of that, you see basically the chemisorbed hydrogen, so the hydrides on top of it. And from the side, it would look something like that. You have the not very reactive H2 being chemisorbed on the surface of the metal or mineral. And this can easily react with CO2 that also can be very well physisorbed, so not completely bound to the surface of the metals or minerals. But also this banding process helps the reduction of CO2 immensely. So we know that in biology, we have these catalysts who are activating hydrogen for us. They're called enzymes. And more specifically, when we again look at the acetyl-CoA pathway, almost any enzyme involved bears transition metals. And that is also true for the ones that specifically activate hydrogen and help hydrogen and the electrons of hydrogen into metabolism. And those are called hydrogenases. And no surprise there, in the active center of hydrogenases, you also find transition metals such as nickel and iron. So there are different types of hydrogenases, but they either have iron or nickel or both. So when it comes to geological systems and geochemical systems, we also know that we have potential catalysts there as well. So a lot of transition metals in different forms. We have just the three examples that we worked with were a nickel iron alloy called everuite, greigite, an iron sulfide, and basically the oxide form of greigite, magnetite, that you all find in serpentinizing systems. And that is what we started with. Just a brief excursion to the process of serpentinization itself. I guess there are probably some people here listening to this that know a lot more details than I do. So please excuse me if I simplify it. But in the end, serpentinization, which is geochemically seen and from the rock side of things seen very complex, can be broken down very simply to the oxidation of an iron 2 mineral to an iron 3 mineral. And on the way, the electrons are being transferred to water that then gives rise to molecular hydrogen. At the same time, this whole process, by the way, is also the process that makes alkaline hydrothermal vents alkaline, because you are left with hydroxides in the end that make the system alkaline. And not only hydrogen is being produced, but also within the serpentinizing process, you produce the catalysts that you could then use or can use for the CO2 fixation part. So not only magnetite, which is a main product of serpentinization, but also side products such as nickel iron alloys are very potent catalysts. So apart from that, we also know that not only methane, but methane is one of the most known and most observed effluents of these alkaline hydrothermal systems. It was already shown that it's from an abiotic source. And so one can assume it's actually the product of a CO2 fixation within the vent system with hydrogen, for example, but it's still not completely known if it really works like that. So you can only assume that within the system, the freshly produced hydrogen can actually react with the carbonates or the CO2 in the system. And we go the step further that we are not only interested in the source of the reduced effluent that actually comes out of it. We really think that this reaction between hydrogen and CO2 or carbonates can actually kickstart something like a primordial metabolism. So with that said, we're not working with actual hydrothermal vents at the bottom of the ocean. I work with these beauties, steel reactors. And our experimental setup for CO2 fixation under hydrothermal vent conditions is actually quite simple. We have these steel reactors. We can pressurize them with CO2 gas and hydrogen gas with pressures up to 25 bar, which translate roughly into 0.5 millimolar of hydrogen gas in solution. And then in these steel reactors, we put small glass vials with a lid on top that has a septum that we slit so the gases can actually go in. And then after the reaction, we do gas chromatography and also, of course, analysis of the supernatants of the liquids and the solid parts for the spent catalysts. And more detail, our experimental workflow for this project looked like this. We had these glass vials with 1 milliliter of water. We normalized our catalysts by the number of moles of metal atoms per compound. So not just the the concentration basically of the compound itself. And we did experiments at lower pH, which just means basically we did not use any buffer or anything else. We just pressurized it with CO2 and got pHs around 5 to 4 just by the dissolved carbonic acid that we had in solution. And if we wanted to have some more alkaline samples, we basically just added potassium hydroxide and thus resulting at a pH between 8 and 9, sometimes 10, depending on the experiment. Assuming that, not only assuming, but you can be kind of sure that the active carbon species here and the alkaline solutions would be bicarbonate. So, and then for both sides, after the reaction, we basically just centrifuged in the ones with lower pHs. We also had to add KOH after the reaction. So you get rid of the iron ions or metal ions in solution. So you could actually submit them to the quantitative NMR that we used, proton NMR, and we used X-ray diffraction to characterize our our catalysts. So this is an overview of the X-rays, X-ray diffraction, and the XRDs we did before and after the reaction of our catalysts. Here's evaruite, magnetite, and greigite. And you can very well see that after the reaction, the XRDs showed no new compounds, so the catalysts weren't spent. The only small difference that we saw was here with evaruite. So we had a small oxidation event, so to speak, with the evaruite, but I will come back to that in a second. When it comes to how did we get to these catalysts, the magnetite we basically just obtained commercially. The evaruite and the greigite both were synthesized by very smart and very able people. On the evaruite side, it was collaboration partners from here in Germany, from the Max Planck Institute for Coal Research, And on the Greigite side, and also the synthesis with Greigite was done by Kensuke in Japan, supervised by Yoichi Kamagata. If you are interested in more details on the synthesis, I'm happy to provide that in the questions or send you the according papers. Just let me know. So to the actual CO2 fixation part that we did with everruite, we just to remind you, we did it under 25 bar, either only CO2 as controls basically, or hydrogen and CO2. And at different pHs. And please also note that this is a logarithmic scale because the product spectrum that we got was kind of different for each product. But all the products that you see here, so formate, acetate, pyruvate, and methanol, were the only products that we saw in our NMRs. So these are really the main products from the simple CO2 fixation overnight. And with only CO2, we still, so without hydrogen, we already got some results. Actually, also not too little formate, especially almost 1 millimole. But that is what I said before. We had small oxidation in the everruite, so we just assume, as it would happen with solid iron, for example, you can produce hydrogen freshly, so to speak. You oxidize metals, you produce hydrogen, and then you can use that hydrogen for the reduction, and that's probably what happened here. But there's a big difference to if we actually pressurize with 10 bar of hydrogen as well. So that's a partial pressure we had here. And then we get formates, especially in better so under alkaline conditions. We got formate over 200 millimolar, acetate around 0.1 millimolar, and methanol over 0.1 millimolar. Smaller, sorry, that is the wrong side anyway. What I did then is basically just went a little bit nuts with the amount of catalyst I used. So I just used more everruite, and the more I used, I also got over the detection threshold of pyruvate. So the next step after the acetyl group in the Acetyl-Coa pathway, which was really exciting as well. And in the end, we got roughly a little bit over 10 micromolar of pyruvate as well. And we also were able to detect small amounts of the complete reduced form, so of methane, which was really nice as well. So next in line was the Magnetite experiments, which were performed by my colleague Kamila Mokowska in the University of Strasbourg. And she could show that magnetite has the same product spectrum, but it has different concentrations. So it's still, those are the 4 main products that we get, but under different concentrations than we get with everruite. So formate was, in comparison with the nickel compound, quite low. But an acetate and methanol are roughly the same, but pyruvate, even at lower concentrations or amounts of catalyst, was quite a lot already. And last but not least, Kensuke also did not only did he make the Greigites, but he also performed the CO2 fixation experiments, and this is just a small fraction of what he did. But he could show, I think in the first time ever, that iron sulfides can actually be catalyst for CO2 reduction in that way, with the help of hydrogen as an electron donor. So he got formate also in quite high concentrations, over 4 millimolar, and acetate around 0.4 millimolar. He was also able to detect methane after 24 days at 100 bar and 60 degrees. So we can only assume what we would detect if we had more time to actually look at our reactions for a longer time, or geological time frames. So to sum this whole thing up, we gave it the title, the abiotic pattern of biotic CO2 fixation, because that is almost the last form of Acetyl-Coa pathway that you will see from left to right, not from bottom to top. You have the biotic Acetyl-Coa pathway here, and here you can see the main products that we got are basically all the steps or the intermediates of the Acetyl-Coa pathway, and even one step more with the pyruvate. So that doesn't mean that's not proto-metabolism or anything. It's really just showing that there is kind of a connection here when it comes to CO2 fixation with hydrogen. And if we think back of carbon reduction or CO2 reduction being a prerequisite for life, it might be that this reaction between hydrogen and CO2 is actually how this whole thing started. And we often got the question about mechanisms on the surface of catalysts. So I wanted to say a few words about that as well. So the thing is, if you think about the Haber-Bosch process, really industrially started in the 20s, even a little bit earlier. And it took over 6 years to kind of figure out how that works on a catalyst. And there was a lot of need from an industrial side to actually figure out in order to find even better catalysts for this very, very important industrial reaction to actually produce ammonia. So we're not sure how long it will take us to figure out what is exactly happening on the surface of these minerals. But we tried to get a little bit inspiration from what we already know from the chemical field, and that usually when it comes to CO2 fixation, goes back to industrial hydrogenation of carbon. So what you can see here is really the final form of Acetyl-Coa pathway that I will show here. Just as a reminder, we have on one side the so-called methyl branch with all the transition metal employing enzymes that we have, with all the different in-between intermediate steps that we have here. And we have the carbonyl branch here, where we basically just produce CO from CO2 with 2 electrons. And then we have the final step to acetyl-CoA. On the other side, if we look at industrial hydrogenation, there is a process that is quite well known already on the surface of starting with magnetite, but the working catalyst within the hydrogenation process is actually something different. But there you already know more or less what the species on the surface of the catalyst are. So we don't know if that's what is happening in our case. We can only assume that it might be very close to what we are observing as well. And when it comes to the carbonyl branch, you have, of course, a reverse water shift gas reaction that can also just be employed here. And even for the final step, industry came up with something, but had actually the acetyl-CoA pathway in mind for this last step, for the so-called Monsanto process, that also basically unites methanol with a carbonyl group to get acetic acid. So that is just how we try to get closer to the truth that we don't know yet about the mechanisms. With that said, I also just wanted to give a quick outlook of what else we are trying to do. So we try to move a little bit beyond CO2 fixation only. And so we're trying to fix N2, so nitrogen as well. And we know that is also possible in nature, of course. Nitrogenases do that. They both split because nitrogen hates reacting even more N2 than hydrogen. They both nitrogenases are able to split both of the molecules in half and make them react with each other to ammonia. And of course, I already told you, and you know that process, the Haber-Bosch process, can do that on a mineral surface, on a magnetite surface, actually, in industry as well. And we know that is basically also nothing more but splitting up N2 and H2 to N atoms and H atoms on a mineral surface, which then overcomes the kinetic barrier, and then they can actually react on the surface with each other very smoothly. What does it mean for a serpentizing system? Well, again, we're far from the natural system here, but we had just this crazy idea that if we mix N2 and CO2 fixation with hydrogen together of course, in our steel reactor, not a serpentinizing system, but under serpentinizing system conditions, we would assume we have hydrogen that is being synthesized, of course, via serpentinization. We have N2 dissolved from the atmosphere, dissolved in the ocean. And both of them can react over catalysts or minerals that we find in serpentinizing systems to ammonia. That was actually already shown that you can at least measure concentrations of ammonia in these systems as well. So our idea is, what if we mix not only N2 in this whole thing, but also CO2? What could we get from there? So in theory, that's what we are picturing. You have a mineral surface with a lot of different reduction products, from both nitrogen fixation and new nitrogen-carbon bonds, and also from CO2 fixation. And they're all basically on the same mineral surface, and then they can actually react with each other because their kinetic barrier is already lowered that way. I mean, you call it activated molecule zoo. It's also far from being proved or anything. It's just the idea we had. And we did some experiments, not starting from N2, but from ammonia, that actually are kind of promising. So we will have to see if we actually get to this thermodynamic stable products at the other end. Of course, it would be great to have not only amino acids, but also in theory nucleobases and cofactors and so on and so on. So we'll see. So this basically covers our fixation metabolistic part of our research. And as I think have a little bit of time, I would just like to share some ideas with you that might also be interesting in an astro context when you look at ocean walls and so on, what could go on underneath these thick ice layers that we're seeing here. So we are always trying to kind of find parallels and patterns that arise in geochemistry that we already know from biochemistry. And as you all know, I mean, otherwise there wouldn't be a Goldilocks zone for the liquid water, right? Life needs water. But from the other side, from the origin of life research, we also know that water is very, very hard to our synthesis. So we struggle with it, because as soon as you actually produce something, if you have it in aqueous conditions, hydrolysis will basically break up your polymerization products or even more complex molecules that you want to produce. So almost any hypothesis out there on the origin of life struggles with hydrolysis. So we thought maybe if we approach it with this biochemistry, geochemistry comparison, we might also come up with something here. So if we look at serpentinizing systems, you would assume, of course, they're underwater, so there's a lot of water, so everything will be hydrolyzed. It's also a big criticism in this whole band theory thing of origin of life. But actually, serpentinizing systems, if you look very closely, more closely at them, could actually provide environments of low water activity, and thus there could be controlled hydrolysis be possible. So because not only mineral surfaces, but also dissolved salts can actually bind water and thus decrease the amount of bulk water you have in these systems. And bound water reacts very differently than bulk water. It won't hydrolyze everything that comes into its way. So what we very simply did in a paper we published last year, we just compared what we see in biochemical systems. For example, we have a cytosol that is full of water, but we have a lot of dissolved molecules. It's very cramped inside a cell, dissolved molecules and salts. So hydrolysis is actually never an issue there, although it is full of water. And in geochemistry, in serpentinizing systems, we know that serpentinization consumes a lot of water, and the concentration of salts and minerals actually does increase during that process. There was a very nice paper, I think in 2019 by Hector Lamadrid et al. That I would also recommend in that context, which actually showed exactly that the water is being consumed during serpentinization, and basically salt will be overwhelmingly highly concentrated inside of the pores in such systems. You have in a cell, you have the formation of micro compartments where you actually can then have small hydrolysis events, for example, where it is in a controlled pocket of an enzyme, you can actually do hydrolysis or even prevent hydrolysis, depending on what you want to do. And for that, in serpentinizing systems, we have mineral pores and micro size and nano size even, that was also shown by a lot of geochemists already. So this is what we have on both sides. And in the end, this both results on both sides to less bulk water and more bound water, and then presumably lower water activity. And both of this, I mean, in biochemistry, you know, it facilitates polymerization, although of course it's also a very simplistic view of a cell. It's more than that, but just for the sake of the argument. And on the geochemical side, we don't know that, but it might. So we have to find that out at some point. I already hinted that a little. In biology, hydrolysis does not just happen. It has to be controlled. It has to be promoted, actually. So there are so-called hydrolysis, or what I'm showing here is a so-called pyrophosphatase type hydrolase. So it is hydrolyzing pyrophosphate, and with the help of divalent metal ions, in that case magnesium, but it could also be manganese. And you actually really need these enzymatic pockets where the pyrophosphate gets in and you have one molecule of water actually doing the hydrolysis in a promoted way. So just a question for all of us is basically, could this be a geological mechanism as well? So with all of this in mind, we thought there might be two possible roles, probably more, but let's just go with this one. Roles of hydrolysis in serpentinizing systems, when it's also in respect of origin of life and polymerization and so on. You have an environment that you have an environment with very low water activity because everything is very salty and very bound to the surfaces. So their hydrolysis would be a very essential reaction that you actually have to promote, for example, with magnesium. So brucite, for example, is a mineral that you also find in serpentinizing systems that need to be there to promote the hydrolysis with the very little water that is at hand. And for example, if you have an environment with a rather medium water activity, so not too high, not too low, their hydrolysis might be even a selective driving force towards more complex molecules. So it basically hydrolyzes the small molecules that are not so stable, but the more stable ones that could more quickly form, for example, could survive. So just for the lack of a catchier phrase, we went with the survival of the sturdy for that, to have some picture in mind. So this brings me to the overall conclusion that micro or even nanopores in serpentinizing systems could maybe serve as a type of protocells with low water activity. And with the salts and minerals we have in these pores, it might be possible. And what we would imagine it like is that we have, starting with very simple reduction reactions that we are trying to show already with CO2 fixation, with nitrogen ammonia fixation, get the first amino acids and more complex molecules. I mean, from B to C, it's quite a jump. To autocatalytic networks would be a possible next step for that, if you actually have a mean to activate molecules to react with each other. And last but not least, you would have something like a ancestor of the last universal common ancestor. You would have mineral assisted polymerization and folding. You would have something very prebiotic, but very close to life already. Of course, that is all imagination for now, but it still kind of connects back to what Luca could have been. So it would be a pre-step to something that we could still kind of reconstruct, to a jump to something that we might be able to reconstruct eventually from the bottom-up approach. And with this, I would like to thank you for listening to my Germanized rambling for the last 40 minutes. Thank you very much again for the invitation, and I would like to thank all my colleagues at Heinrich Heine University, and of course, science would not be what it is without all the collaboration partners that we had in the Max Planck Institute in Strasbourg, and also in Tokyo and Sapporo. So thank you very much. And I stop.
Thank you very much, Martina. It was a really, really great talk. Do we have any question. If you have a question, please use the raise hand button. Okay, we have several. Estelle, do you want to start?
Yes, hello, thank you for your talk. It was really interesting. I'm not sure I understood how you account for the pH and thermal gradients in the experimental setup that you showed, because these are definitely like key in hydrothermal vents. So I was just wondering.
Oh, that's a very good question. I actually just mentioned them in my talk. That's actually completely true. And then I ignored them because we never do them. Because a lot of people and researchers, also friends of mine, they're working on pH gradients and hydrothermal vents for a long time already. And I always think that pH gradients will be very interesting at different points of complexity rather than what we're doing right now. Because for CO2 fixation, for example, we really don't need it. We just need the hydrogen and it works to a certain extent. So I think to actually harvest pH gradients, I mean, that's just my opinion. There are a lot of people who disagree. You would need a more complex system already.
Okay.
So that's, yeah, I have it there. That's true, but it has nothing to do with my experiments.
Okay. Thank you.
Vlad, you have a question as well?
Yes. Hello. Yeah, thanks for a very interesting talk. So I have three questions. So the first question is about the what is the rate of reduction of CO2 to CO, and how it can be compared to the photolysis of CO2, from the UV emission, from especially from the young sun. So how it what is contribution? What is its contribution to the formation of CO to CO2 ratio?
You mean in our experiments or in general?
Well, yeah, what rates you can get from your experiments as compared to the what can we expect from the CO2 photolysis rates?
I actually don't know. I don't know the exact rates of the UV catalysis, to be honest. Nevertheless, I also cannot tell you a lot about our rates because we never measured CO in our gas headspace. We only got methane. I mean, we repeated them repeatedly. We also had some CO, that's true, but it was very fluctuating. So we always kind of thought we were definitely producing CO on the way. Especially with the nickel minerals, because we got a lot of formate, and that is the natural intermediate for this on a surface. But I really cannot tell you the kinetic rates. But we actually have now a new PhD student who wants to do more kinetics on our experiments anyways. But that was not the main thing we were looking at. But very good question. I wish that at some point, maybe the next 2 years, Max, that's his name, can actually provide you with more answers than that.
Mm-hmm. My second question about that, you talk about nitrogenous species, and and of course the hydrogen cyanide, one of the major players in the whole prebiotic chemistry, right? So the question that how would you produce hydrogen cyanide in this scenario in the hydrothermal vents?
Oh man, I actually would love to say everything in this question, but it's actually unpublished. But in theory, the thing is, HCN is the anhydride of formamide, right? So and formamide you can actually produce very easily if you have enough formate and you have ammonia, then you get formamide. The thing is, HCN is not what we actually want to have because it has really not a lot of connection to biology. Nevertheless, it could be produced if you have at least, I mean, HCN itself might not be being produced, but it's hydride, if you want to call it like that. Yeah, sorry, English. So formamide should be easily produced in these systems, which is one step closer.
Yeah, and the last quick question is about the so yeah, you talked about the micropores, and so what is the role of wet-dry cycles, and how can you create wet-dry cycles, because they're extremely important in formation of the biopolymers, right?
Yes, thank you so much for the question, because I actually forgot to say that in my excitement. That is exactly this paper that I said, the Madrid et al. In 2019. They actually showed. I mean, they don't call it like that, but if serpentinization consumes a lot of water, the whole system dries up. But then a new wave, so to speak, of ocean water gets into the system. So you actually get wet and dry, or presumably you can get wet and dry cycling inside these vents, which is, if we could actually prove that, eventually would be a game changer for the whole thing, because then we can actually do wet dry cycling within these systems as well. And we have still hydrogen. So that would be very great.
Yeah,
but yeah, that's exactly the point. Sorry, I forgot to say.
Sure. Thank you, Vlad, for the great question.
Thank
so do we have any other questions? I see that Max, you have written a very interesting comment in the chat. So do you want to speak up?
The question is there. It's just about the idea of temperature variation, perhaps, as a way of dealing with the hydrolysis, or the water cycling through, out of, and then back into the hot zone, something like that. I was just wondering if you had any notions about that.
That's a really interesting point, because it actually comes down, if I understand correctly, also a little bit to convection, right? So you would have, for example, within a pore, you could have a hot side and a cold side, where you could actually condensate respectively polymerization products, for example. So I think when we talk about gradients within these crust systems, you have the big thermal gradient. So you start hot and then it gets colder and it gets easier to condensate and to collect and stick to the mineral surfaces. But I think you also have kind of tiny convection heat gradients inside the pores that could help with that. So it could definitely be it could play definitely a role as well. So the one thing is always with these things, it's super hard to do that in the lab. There are people who are looking at, I think, Dieter Braun's lab in Munich, and probably a lot of other people that I don't necessarily know are trying to do this convection, for example, to observe what you can actually do with it, but to have a complete hydrothermal system there that you can just basically look at and have the right thermal gradients and then just take out your products in the middle is still a little bit far away, unfortunately. But if I could build something like that, I would definitely try to have these thermal effects there as well.
Thank you. Estelle, do you have another question?
Yes, I was just wondering, since hydrothermal vents are usually deep sea systems, do you insist on saying serpentinizing systems so that it does not include only deep sea vents, or so what other systems are you thinking about?
I mean, it's also a great question. Thank you very much, Estelle. So that's where my PI and I go separate ways sometimes, because I really insist on the serpentinizing systems, because I think as soon as you have phyolites or even more shallow serpentinizing systems, this could still work. So we don't need the high pressures of the deep sea for that. So I mean, also look at the pressures we were doing at 25 bar. That's not that deep in the sea, right? And I think the partial pressures of hydrogen that you would need, like 10 bar, maybe 5 bar, you can also get on very shallow serpentinizing systems. I mean, you still need the water to actually produce the hydrogen, but I personally would not insist on deep sea vents when it comes to that. So
okay,
as long as you have the minerals and the hydrogen and the carbonates, I think anything is possible.
Great, thank you.
Thank you, Estelle. So I have the two naive questions because it's a bit outside my wheelhouse, but so I think for life, right, you have the two different approach, bottom up and top down. And if I understood correctly here, you favor the top down approach. And so can you describe what is the other one, the bottom up, and why do you favor the top down?
Oh, I don't favor any of those. I think that both of them are very important, and one could not really live without the other because both of them have problems. For example, I sometimes think that a lot of the bottom-up things in Origin of Life are very far away from biology, which is fine. You can do whatever you want, but I need the connection to biology because life as we know it is basically what we see here on Earth. So we have to have a connection to that somehow. Otherwise, we just do anything we like and then label it life eventually. I don't know. But that's also just a personal view of things. And the top down, for example, also has its weaknesses because it's also just a reconstruction based on genes. It also is basically someone writes a code to compare stuff, and it has to be anchored somewhere as well because it also is quite abstract in many ways. So I think only when we do both, and even there are some middle out ways in Origin of Life theory as well, that's how we get at least a little bit closer to solve this if we ever get there. But I think really we need both. The top down is really just what biology has to offer, and I think it also reminds us where we come from when we ask this question. That's why I think top down is also very important here.
Okay, I see. Thank you. And yeah, my other question was about the LUCA. So does that the properties that we expect from LUCA have to be really specific to Earth environment, or it's so at the root of life as we know it, or even of life as we don't know it, that it can be something more universal?
Yes, definitely. I mean, the thing is, in the end, when you just look at this reconstruction and any system where you would find these, I mean, that's the thing with the top down. So we're really bound to the physiology that we see from the reconstruction. If we want to have something more abstract, we would have to start somewhere differently. But when it comes to if it really originated in hydrothermal vents or something like that, in the end, any surrounding that provides metals and hydrogen and some carbon source should also suffice no matter where it is, I guess. So it does not have to be that. If I understood your question correctly, I'm not sure.
Yeah, absolutely. That's very cool. Thank you. Estelle, is it your hand is still raised? Is for
yeah, I just have a final very quick question. There's also a question in the chat, but yeah, very quickly. You mentioned that there were some hypotheses where hydrolysis was not a problem, and I just have no idea what you had in mind.
I mean, that's basically the whole wet-dry cycling thing, where you have, or I mean, from a prebiotic chemical point of view, as soon as you are on the Earth's surface, you don't have a lot of water unless you actually want it there, like in ponds and so on and so on. And there they create an environment where water is not a problem. That's what I meant. Okay. I mean, if you have too much, of course, it will always fail. But if you look at this typical pond surface, wet-dry cycling on the surface, I mean, water is there when you need it, and it's gone when you need to polymerize.
Yeah. Okay. Thank you.
Thank you.
Thank you. And yes, we have a question in the chat from Dave. How about surface chemistry on dust grains in protoplanetary disks? Are these grains viable candidates for the chemistry you are describing?
Well, there you have to help me a little with the background because it's a little bit far off. But I mean, I read a few papers on that, but I'm really not an expert on that. But let me just say, if you have any kind of mineral surface or metal surface, and you have certain gases in that whole mix, like hydrogen or nitrogen or whatever gas you can think of in a protoplanetary disk, I can definitely imagine that could happen there as well, depending on, of course, the temperatures, but that's what I don't know. But if it's not too cold and you have some kind of an external thermal energy source, it should I wouldn't see why not. But that is a very layman answer because it's really far off my comfort zone of knowledge.
I can add a little bit to that, if you can hear me.
Yes.
So a protoplanetary disk is expected to have structure to it. We talk about a snow line outside of which the water is frozen. That would be like a mid-plane snow line. So if you picture the disk as having kind of a flared structure in the direction perpendicular to the orbital motion, there's a flare to the disk shape. Within the mid-plane itself, there's a snow line. Inside of that, water would be evaporated essentially, and outside of it, it would be frozen onto grain surfaces. But within the disk, the orbiting disk that has that flare structure, the deeper you go into the disk, the more frozen water would be. But you would expect to have some kind of circulation of the grains within the disk. In the vertical sense, and the closer they get to the surface, the more likely water would be to be exposed, well, the grains would be to be exposed to the heating from the star. So at the very surface, water could evaporate from the grains, but close to the surface, but still within it, you might expect, you know, a temperature gradient and more frozen water as you go deeper and deeper.
So could there be like a freeze and condensating, so melting cycle kind of thing?
I think that would be plausible. Mm-hmm.
So, I mean, that would go very close to what also Max, I think, suggested before.
Mm-hmm.
So, I mean, if you take into account that temperature can definitely also have an impact, not only on the reactions itself, so the reduction reactions, but also maybe on polymerization. Again, I don't see why not, especially because then water is even less of an issue because it's just so, I mean, it's very briefly there and then either evaporated or frozen again. So,
right. And I phrased the question in terms of dust grains, but as time evolves fairly quickly in the early development of a protoplanetary disk, you get the formation of planetesimals and larger chunks of material, things that are more comet-like. So larger bodies, but still, you know, an inner rocky core with potentially a water-like surface or water intermixed.
Yeah, so that means you would just basically increase or actually decrease the surface you have, but you could still have these kind of reactions, I guess, on these surfaces as well. I actually never thought about that because I always thought about porous systems, you know, and you always need to have water coming in a, you know, I actually forgot the name right now, so that's where you can see that I'm not a geochemist. But when water comes in, subduction, right? So, but it might also work without this principle as long as you have systems that work with that. But that's a really interesting thought. So thank you for that.
You're very welcome, Martina. Thanks. It was a great talk.
Thank you.
Thank you, Dave. So we'll take a last question. Vlad, go ahead.
Yes. So you have the question about the about the scenarios, hydrothermal scenario versus the small pond scenario. And the point is that you said that the hydrothermal vents can be pretty close to the surface. Yeah, that's very important to have this dry wet scenario. But what's more even important, I think, is the concentration of those molecular species, right? So we need to get to the point of saturation in order to start polymerization process. So that's why we need those small ponds rather than big ocean where everything will be quickly dissolved and you never will have the required concentration for polymerization. How can we reconcile this?
I actually think that the ocean, even if you are in a deep ocean, it would not be an issue because actually the pores themselves that are basically coated with minerals are already basically like chromatographic traps where you can concentrate certain peptides or amino acids. I mean, let's not get ahead of ourselves, but I'm pretty sure that it's not about the outside of the system that much. I just want to make it, when I say any serpentinizing system will do, it's basically because I think it could have happened in a very broad sense all over the Earth. and 4 billion years ago. But I really think it's not about where it's collected outside, but rather in the system. So I really think those nanopores, I mean, if you look at them also nowadays, there are also publications showing that they're clogged with stuff, very like with the amino acids and like super complex, even chondrite-like things, so that came there abiotically. So, and they had to come from somewhere. So I guess actually the pores themselves are this pool, this pond, and not the ocean or the pond outside of the system, because
So
they're also protected there.
Yeah, so those pores can serve for the hydrolysis of to basically to polymerize the amino acids into peptides.
Yes.
So, yeah, interesting. Okay, thank you. So I need to think about it.
Yes, I mean, I would be happy. I mean, Thomas, you can also share my email, or I mean, you can just Google me. But whenever there's other questions or discussion points, please feel free to send me an email.
Thank you, appreciate it.
Okay. Well, so it's now past 12. Thanks again, our speaker, Martina. Thank you very much for your great talk. And we had a lot of questions, very interesting questions. Also, thank you everyone for that. And so, yes, our next Exoplanet seminar will be on June 24 by Tessa Fisher, and it would be about what atmospheric chemical reaction network topology can tell us about exoplanets. So it's going to be also a very interesting talk.
Okay.
So again, thank you, Martina. Thank you, everyone. And
thank everyone.
see you next time.
Goodbye.
Thank you. Goodbye.